Yeah. My heart is happy that we've gathered here today in this place. So welcome. Thank you all. Kaya Wanji.
Thank you so much, Mitchell. I've had the pleasure of being welcomed by Mitchell at a few years of planting days up at Clontarf, and also at a corridor yarning event hosted by Adam Peck earlier this year. And it's always so beautiful and so powerful. So thank you so much again for welcoming us here this morning. We're all really happy to be here. So we've had a slight program change this morning with Jack unfortunately being able to be here. Thank you, Eddie. And she sends her apologies. In her place, the team will be sharing the MC role this afternoon. I'd like to welcome our online audience via Zoom, and just a gentle reminder to please keep your mics muted. Thank you so much for joining us this afternoon. We are recording this afternoon's forum, and you'll all receive a copy post-session. I'll be looking after the tech. My name's Eve. I'm the Member Services Manager here at WALN. Vanessa Slater, our amazing Landcare Expertise Exchange Facilitator, will be introducing our guest speakers. And we're delighted to have been joined by a new intern from the University of Notre Dame, Georgia Nichols. She's just over there. She'll be helping me keep the speakers on time this morning. If at any time you need help throughout this afternoon, please don't hesitate to reach out to one of us. The emergency exits are just out this door, out the front door, and if you could just head up to the front car park. So before we kick off, the shape of this afternoon will be 4 presentations, each following closely behind the other. So if you could please note any questions for each speaker along the way. After we break for afternoon tea, we'll come back in and we're going to have a structured workshop, kind of like a Q&A session where we'll break into groups of 4 groups, and each group will have 13 minutes with each speaker. So if you find you have more questions than you have time to ask, please don't stress, simply email them in. Our online audience are welcome to do the same. Ness will also be developing a resource kit for the WALN Knowledge Hub, and all of the presentations will be available there too. So after the final presentation, we will break at 2:30 p.m. for some afternoon tea from All Good Grub, the amazing First Nations caterers. We will have our workshop and we'll aim to finish by about 3:55-4. Please feel free to continue your discussions at the front of the venue. But we'll have to make a relatively swift departure around 4, as there's another event here afterwards. So thanks again to the speakers for their time, to Melbourne City Council for the use of this great venue, and to all of you for coming today and for your interest in this topic. So I would love to introduce our first speaker, Julie Fielder from Phytos Environmental. Welcome, Julie.
Yeah, I've tried to condense down a little bit about fungi and restoration planting down to 25 minutes, but it's been a bit of a challenge doing that. So what I'm going to do is explain the ecological side of things here, and then Eve's going to be able to email you all out the management action slides at the end, because it just wasn't time to get through the whole lot without having some of the preamble people need for not knowing a lot about fungi. So yeah, I just want to acknowledge country. Thank you for welcoming us this morning. Yeah, and we're meeting today at Biddy here at Piney Lakes. So why should you care about fungi in your restoration plantings? So that's what we're going to explore today, is the basic ecology of what's going on. So as most of you are aware, we've got a restoration continuum. So it's a nice diagram out of our new book, Fungi for Land. So what we're trying to do is move a dysfunctional ecosystem that's missing large amounts of these functional groups and ecosystem processes back in along that continuum through to a diverse functioning ecosystem. So we're trying to get more native species, more of the functional groups that are coming back in, more of those processes by similarly to plants, fungi work in quite a similar way, and what we're trying to get back. But as we'll see, it's a little bit more complicated than that. So fungi provide a huge amount of ecosystem services that a lot of people don't really understand, often quite overlooked. So absolutely integral for decomposition, particularly lignin and celluloses. Mycorrhizal association, so they're that partnership that we're forming between fungi and plants to allow nutrient exchange. They're really important for cleaning water in our waterways. In groups called the Ingoldian fungi actually take out a lot of the toxins and pollutants that we have. Absolutely important for nutrient cycling and availability to our plants. They can also accumulate heavy metals. We've got like metal toxicity in sites we're trying to restore. And they're really important for creating that soil structure that's so useful, so important for water percolation and retention as well through separation of quality. The fungi are involved in all levels of soil formation. They're involved in the weathering of parent rock, so they'll get in with their mycelium and drill them down into the rock, split it apart, and also exude enzymes and actually eat the rock. They are involved in biomineralisation, which is partly that they excrete things like calcium oxalates that cause sedimentary rocks to also be formed. So there's a good relationship with the geomorphology there. Nutrient cycling, stale soil carbon aggregates, nutrient exchanges with plants, and the piping that you can see here running through the soil with fine filamentous strands of what's actually stabilising the soil surface. So really important for metagenesis. You won't have soil development unless you've got an active fungal community living in your soil. So plant growth, which is what we're aiming for when we're doing a restoration, requires nutrient acquisition and transfer, which requires fungi. So all nutrient cycling has a biotic component. Fungi are particularly integral in that in this. Bacteria and archaea will also help with nutrient cycling, but if you're wanting large amounts of carbon transport or large amounts of phosphorus and nitrogen moving around, you're really looking at having to have those fungal partners with your plants to make that happen. And fungal hyphae are also really important for tying trees together and allowing nutrient transfer from trees to the soil to the trees, but also between trees as well. So that's that binding network that's tying your restoration site together. So I thought I'd do a quick overview on the main groups because I'm going to use some of these big, nasty, long words, and I'm going to do that because I don't have any common name equivalents. So we're just kind of stuck with them at this point. So got 19 phyla of fungi. So fungi are their own kingdom, separate from plants and animals. Next stage down in the taxonomic classification is phyla. So we've got 19. The main ones we're going to talk to today are the Basidiomycota. Call this because they have spores on Basidia. So Basidia is this structure up here. And you can see the spores connected to the end of it. So it's got that kind of reproductive structure, Basidiomycota. We've got Ascomycota, which is our most diverse group of soil fungi that we're going to get. And these are called, they're an ascus, and ascus is a sac. So we can see this beautiful morel, Morchella elida here. We've got these nice spores in the sac here. We've got endophytes. So these live on roots and leaves and other parts of the plant bodies. And for at least part of their life cycle. And they can work with other fungi to increase plant health. So they're like little fungi that hang off your leaves and roots. And then we get entomopathogenic fungi, which they're down here on the left. These are fungi that come along and kill insects. You would have seen things like zombie ants and stuff probably on the news or in the Curio kind of pieces. They actually are really, their asexual phase is really abundant in the soil. And they enable plants to ward off insect attack by producing insecticides that they channel into the plant. And they also kill animals in the ground and provide nitrogen to the plant through that as well. So, yeah, we've got some nice entomopathogens here. Got some nice endophytes where you can see the fungi actually growing into the cell on the leaf surface. So we've got a lot of different niches happening within our restoration plan. The types of mycorrhiza. We're discovering new types of mycorrhiza all of the time. The two ones I want you guys to try and remember are the two main ones is the arbuscular mycorrhiza. And they're called arbuscular mycorrhiza because they make little tiny tree-shaped organs within the plant root cell. So, and that's in the glomeromycota. So nice, tidy little group to remember. And then we've got our ectomycorrhizal fungi. These are our relationships we have with woody trees and woody shrubs. They can be from either the Basidiomycota or the Ascomycota, but a lot of the ones that you'll see fruiting around the place are going to be those big Basidiomycota, your big mushroom fungi and big skin grass fungi that you'll see in the forest that you'd probably all be familiar with. So we also get ectendomycorrhiza, which is sort of a bit on the outside and a bit on the middle. Orchid mycorrhiza, which are a rainbow of fruit flavour and have a lot of different variation, but they generally create these organs called pelletons that are sitting in here, so like little hypercoils within the plant cell. We get our ericaceae, all our heath species have specific types of mycorrhiza as well that we're just still learning about. Endomycorrhiza and the mycorrhiza pine root enterprise, which are our molds that relationships with plant roots uptake. So we've got all of these different groups of fungi. Rob, do you want to see? This one. Yeah. So we've got a whole bunch of different relationships going on, and we're discovering new ones all the time. So every time I log in and read some more papers, then someone else has discovered another type. But today we're just going to plug in with the arbuscula, and then the ectomycorrhiza, which is on the heart and sheath around the roots of the main trees of plants. And I just really wanted to highlight, this is a beautiful slide from Mark Bunjett's mycorrhiza website, which if you haven't explored it and you're interested, it's a really great resource for learning about mycorrhiza. We're really lucky to have Mark as part of our professional community here in Western Australia. He's one of the global experts on particularly on particular mycorrhiza. But he's got a nice picture of a Thyssinotus triandris root cross-section. And we've got this beautiful fungal hyphae growing around the outside, and we have no idea what it is or what it's doing. So it's just to illustrate that we know a bit about what's going on with fungi, but there's a huge knowledge gap in pretty much everything I'm going to tell you today as well. So it's just a big disclaimer on that one. Things might change, and the science is slowly moving forward despite not having many mycologists. So we have some plants, like about 80% of land plants form mycorrhizal relationships, but not all of them. So when we're looking at our restoration plantings, it's worth having a bit of a think about what sort of fungal relationships are plants engaging in. So arbuscular mycorrhizal fungi is your main relationship, forms relationships with the most amount of species of plants. So we've got our poaceae, our grasses, our peas and acacias, our daisies, and our myrtaceae all form arbuscular mycorrhizal relationships. This is a long way from an exhaustive list. If you want an exhaustive list, please go and see Mark's website and learn heaps of it. Our ectomycorrhizal plants are generally woody plants and shrubs. So things like our myrtaceae, our eucalyptus, trimias, beckias, roundnaceae, cryptandra, spiridium plants like this. We get some non-mycorrhizal plants. So our brassicaceae, like our lepidiums and our weedy radishes. Our proteaceae, they've got a totally different root structure, and they're not going to be forming these fungal relationships in the same way. Sedges and rushes, so cyperaceae and juncaceae also are not forming those mycorrhizal relationships. We have specialist mycorrhiza for our orchids and for our heath families, so our stiphilias, leucopogas, and conostephiums. And our fine Mucoromycota, fine root endophytes, so our little mould friends, they're forming relationships with our liverworts, hornworts, and some of the angiosperms. But as I said, these are pretty recently discovered, and we're still learning about them. So we can think about fungi of having three main patterns in restoration. We've got some early stage fungi that are decreasing in abundance as we move along that kind of sequence. As our restoration gets older, they're going to come in early, but they're going to drop out over time. We've got some late stage fungi, so they won't be around in our early restoration, but as the system becomes more specialised and plants become more established, what we're hoping is to get those late stage species back in to support our restoration in moving to that later cereal stage. And then we get the multi-stage fungi that don't really have a distinctive pattern and might, fluctuate and grow depending on other conditions. So, and look, there's a lot of things that play into how fungi are distributed as well. So your past management history is going to play into what's able to survive in your system a lot. Your soil chemistry, so how much phosphorus and nitrogen is remaining. Maybe you've had it's been an agricultural paddock or something like that. That is going to influence what fungi are able to stay. So this is super wordy, and I'm sorry. I've looked at a whole bunch of studies this week. There's not a huge amount done at the moment in Australia looking at a restoration kind of sequence over time for what's actually growing there. So I thought I'd start off introducing you guys to some of the different species we're getting at different stages, but I just really want to bring home that fungal systems are highly complex and highly diverse. So often we're having a huge amount of species, and I've just cherry picked out a few of the ones that we're getting a lot of, but if you think there's going to be thousands of entities at any one of these stages. So that's the other thing with learning about about fungi through high throughput DNA sequencing is you get so much data pulling out big patterns out of it is quite tricky just because our brains don't really cope well with looking at thousands of species at a time, which is what we're doing when we're looking at sort of data as mycologists. So the main thing I want you to have a look at, you have a look at the ascomycota. So over time in this study, so this is from the Mount Lofty Ranges in South Australia, our ascomycota are decreasing as our chronosequence gets older. Basidiomycota are increasing. And then we've got the glomeromycota, which we were talking about, AMF, are decreasing through the chronosequence. Rosellomycota, which is a really base lineage fungi that have little flagella and swim around in the soil, they're decreasing. And then, the other groups you can see, Dothaomycetes, Uromycetes, Lemosomycetes, they're all ascomycetes that often kick in an early stage. We'll still have them in later stages, but their numbers won't be as abundant. We'll start moving into the bigger basidiomycetes that are coming in as we move through. There's another study that I found from Canada. So you can sort of see this is looking at ectomycorrhizal succession over time. So this is like a group of forest scientists. You might have heard of Susan Stimmard. She's the woman who does the Wood Wide Web. So this is one of her research projects. And you can see at 5 years age class of the forest, it's really dominated by this Rhizopogon vinicola, but as it ages, you can see there's a reduction in those species and an increase in things like rustilis and Putnera. So we're really looking at a shifting dynamic as a forest ages, change in reliance on species in different seral stages. And I've definitely seen that supported by eDNA data that I've collected. Older trees will have a different microbiome than younger trees, and they'll have a different species complement. And similarly to vegetation, it gets really old growth vegetation tends to have a simpler structure. There's less diversity being expressed, less plant species. Similar patterns are happening underground with the fungi as well. But we end up with these really well-developed ecologically functional spaces. So yeah, when we have cleared soil, what we're ending up with is a whole bunch of small ascomycetes kicking around doing fairly general things. So these things will be often quite globally distributed and quite simple organisms. You can see they're all quite small, the ones I've put up here. So we've got Chloridium, which I've seen kicking around in the bush a little bit. You get tiny little hairy black things on wood that you can see as the fruit bodies. We've got Cladosporium, which is also known as Davidiella. A lot of these fungi have a sexual and asexual stage, so they will have two scientific names because we didn't know that they were the same organism for a long time. So it is a bit of a mess trying to understand. You've got to work out what the telomorph and animal name is and realise they're the same thing. So common environmental fungi, they can be endophytes or epiphytes. You get heaps of these. Pacillomyces. Some of you might have seen a little tufty white fungus cracking out of an insect kicking around Pacillomyces tenuipes that we get in Western Australia. What we find is we end up with the asexual phase of these kicking around as well. They can be plant pathogens, entomopathogens, so it's ones that eat insects, and also they can kill the nematode traffic fungi. So they do a lot of killing, but they're really widespread and common in things like your compost heap and areas where you're undertaking that breakdown at home. We also get a Cladophiloflora. These are really interesting. A lot of the time when you Google them, you will get medical papers that tell you about how they're going to rip your toenails apart to cause your arm to fall off. But really common in pretty much all soils. And they're probably undertaking some general soil function. But because humans are very human-centric, we only find out about them when they start attacking us. And they do that pretty regularly. But they're probably just a generalist saprotrophin or a plant endophyte that opportunistically attacks animals and humans. So, but yeah, same thing. When I pulled out soil data, there's always been a lot of this. And you're like, where is this from? And they're like, a koala's nasal passages in Kobe in a zoo it's been sampled from. And you're like, well, it's all through my soil on Mount Wellington. Like, what am I doing with this? Yeah. So yeah, like with really simple organisms, lots of ascomycetes, not many basidiomycetes in this cleared soil, very generalist cosmopolitan species. Okay, so then when we've got degraded soil, the first thing that happens when we want to start stabilising soil is often we'll have the formation of a biocrust, particularly if we're doing, say, mine site restoration or something like that in Western Australia in the arid zone. We'll end up with algae, lichens, mosses, liverworts, and fungi, and they will bind together to create an ecosystem that stabilises that soil surface initially. So they cover the soil, they grow really quickly and vigorously, and they're able to decompose lignin and bits of wood and release that nutrients back to the soil. So they're the things that keep starting our restorations as far as getting that soil active. They structure the rhizosphere microbiome, so that's the microbiome that sits around the roots. They start to engage in helping that get organised and start to work. And they help seedlings to adapt to environmental stress and also toxic elements like heavy metals. So some of our sites might have a legacy contamination issue through being close to industry or having a contaminated site. So these are the organisms that are going to make it vaguely hospitable for our seedlings to start surviving. So some nice picture of some Phylobasidium fungi in culture and a nice soil crust here, and then don't bust the crust. So if you've got soil crust forming, try not to get your workers and volunteers to jump all over it because it is doing a really important job in the system. So you want to be limiting that traffic through your site, keeping those crusts intact as they're forming. So early stage, well, we've got our biocrusts, and we've got soil stabilising species which have an ability to access sugars with limited plant species. So they're going to be early stage small gannet. A gannet is what we call an individual fungus. So some of them will have little tiny spreads of mycelium that might be this big, and our late stage one can be up to 300 square metres in size that they're occupying in the soil. So we're starting off with this little small things like a herb. If you imagine them a bit like a herb flowing into the site, little fungus. And we end up with a myriad of small ascomycetes. So some that you might commonly see starting to form is things like lichen omphalia. And this is actually a basidiomycete lichen. So if you have a look, you can see it sitting in this beautiful algal mat at the bottom. So the green stuff covering the soil. So this is actually a lichenized mushroom. And you'll often see these alongside tracks in the bush where they're starting to re-stabilise the soil where there's been disturbance, or in early rehab as well. And then you'll also get things like Leucaria, which grow in big flushes, which I'm sure a lot of you would have seen come up plantations pretty quickly as well. Early stage EMF accumulates heavy metals, not too choosy on who are partners with, happy to make friends with most plants. So it's a really good thing if you're starting to see these in your early stage pick up. You can often tell these because a lot of them have flesh-coloured gills too, so they're nice and easy to spot. Early to mid-stage, we'll start to have things in our basidiomycetes, things like Gallerina, which is this one in the moss, and moss symbionts will start to come in. Your Leucaria will keep kicking through. Scleroderma, which is a really great generalist ectomycorrhizal species, which is this one up here, these puffballs, black middle with the top sort of breaks open, get that nice black spore come out. So that'll form good relationships with your eucalypts and other myrtaceous plants. We'll have things like Tomentella, which is the middle photo up here, and our thellophores will start coming in. And in your ascomycetes, you might start seeing things like earth tongues, our geoblossoms, or exophialia, which is beautiful dark septate endophytes that I've put up. Again, the most common known exophialia is Aquinas because it attacks horses' hooves and causes foot rot. So doing really important things in the soil and looking after our plants, but again, we only know it because it attacks animals that are commercially useful to us. And at this point, we're likely to have mosses, annual plants are starting to reduce in our rehab, our perennial plants are starting to bulk out, and our shrubs are starting to reach height, and the trees are coming up, and we've got this stuff coming in. So when we get to the mid-stage, our basidiomycete groups are becoming more diverse and more specialised. So these are things like our beautiful Cortinarius cynapicolor. So you can see the beautiful cortina on this one forming. That's why they're called Cortinarius, because they have a cortina. We get things like Lactarius in the rusty lilies, and also Inocyphe. This is Inocyphe Bruneo fibrillosa, which is a really common one in Western Australia in Carrier Jarrah forest. And our ascomycetes are going to start to decrease in number and decrease in their importance to the system, but there's still going to be a lot of them. So ascomycetes are always going to be your most diverse group in any sort of soil that you get. And then groups like the archorhizomycetes, which translates to old root fungus. So these hang out in the rhizosphere and in every soil, really functionally important. We're still working out exactly what they want to do, but they start to really pick up in that mid-stage succession. And at this point, we're starting to get a developed overstory. It might not be completely mature, but we have good established trees. a well-developed native ground story, and we'll have herbs and annuals that probably be less diverse but still present. We've lost that diversity curve we get initially after a disturbance, and it'll be starting to drop off. So underground things will be doing a similar thing. Our larger fungi will be starting to kick through, and the little early stage get ones will be starting to drop out. So when we get to late stage, we'll have less species than a mid-stage development. So we find as the diversity's dropped off, but we've got this climax community that's dominated by these big basidiomycete ectomycorrhizal fungi. So these are things like our russulas. So I've got a nice picture here of Russula sanguinea and Russula nearame here. And you'll often find with old trees, they'll have like several species of Russula they'll have a relationship with. These are the ones that get to 300 square metres and are tying that forest together. And that's the climax community we want to be aiming for. We've got that functionality of the ecosystem. We've got that flow of nutrients and moisture through the system between trees with those fungi. Yeah, so we'll also get other things like our piloderma, our corticioid EMS. So sometimes you might see piles of crusty things all tied together with like a white crust in the bush. That's our pilodermas. And our coral fungi as well will also start coming through in abundant numbers in those like old functional systems. So again, I want to stress this is just a snapshot. Every one of these systems will have thousands of little species occupying a lot of ecological niches. The other thing we find with fungi is what you get here, 20 centimetres away under a different shrub with different set of roots, you'll get a different, slightly different composition of fungi. And a lot of fungi will adapt to specific understory species depending on how much phosphorus, calcium, nitrogen that plant A needs and B puts out in its leaf litter fall. So every different type of lignin, hemicellulose and hemicellulose probably have a different type of saprotroph that'll come in, have a preference for those food types and be breaking that down. So there's a whole different diversity of niches within the system. And so what we're trying to do in our restoration is represent as many of those different niches as possible so we can get those species back, come back in and create habitat for them. So this is a really nice paper that someone's done overseas looking at a eucalypt plantation. They inoculated the seedlings with a glomus, which is arbuscular mycorrhizal fungi, and with Physolithus tinctorius, which is that horse dung fungus you would have seen growing along the roadside. So that's a really fairly generalist ectomycorrhizal fungi that likes hanging out with eucalypts, really helps them grow. So you can see it's really dominated by the AMF in the initial sort of 30 to 60 days. And then the ratio is dropping off and we're getting that increased like colonisation by the Physolithus. So the AMF is super important for early stage seedlings, but they're trying to move to those larger fungi as the trees get bigger and more established and find those ectomycorrhizal partners. Someone else has also had a look at the seasonal change because fungi tend to change what they're doing with the season. Because they're very moisture dependent. In the winter, you'll get a lot more mycelial growth. In the summer, it'll dry out and they'll start to bring their mycelia back. What we find in these eucalypt plantations is that they both colonise the same root systems, the AMF and the EMF, in older trees, but the dominance of these species will change with the climate conditions. So in summertime, when the soil's dry, the AMF will dominate and the plant will be very reliant on them. In the wintertime, when we've got those nice moist soil conditions, it'll be relying on our Physolithus rather than our glomus here. So I guess the question I've got, what do we think is going to happen with climate change in a drying environment where our fungal dependence for our eucalypts in Perth? Are we going to switch to a more AMF dominated uptake? What does that mean if they're smaller organisms and not forming the network? So I don't know what the answer is, but it's a pretty good question when we're thinking about resilience within our restoration plantings. There's a nice paper here from Victoria where they've had a look at different revegetation techniques and mycorrhizal colonisation. If you have a look, we've got AM colonisation, they've got their native reference sites quite low. Their seeded treatment, they've got middle kind of colonisation, and their tube stocks very highly colonised by arbuscular mycorrhizal fungi. But in a native situation, we've got species being heavily colonised by ectomycorrhizae, our seeded treatment being middle colonised, and our tube stocks not really forming the relationships the same way. So what they were suggesting is if you want to have like that serial development over time, maybe using seed might be preferential. to using cheap stock to get that microplastic pollinisation in. I know it's not always practical, you know, planting days are easy to take cheap stock and put it out, but it might be worth having a think about, okay, so if I'm using sterile media in a nursery, is that interrupting my pollinisation rate? Put it out. The answer seems to be, yeah, it does actually change things. So, so I guess the big thing thinking through the restoration space is how do we get these fungi back into the system? There's been some ideas about playing around bringing spores in, which I think might be useful using animal scats or collecting fungi from the native, local native bushland, fixing it up, shaping it, putting it in. A big barrier for understanding the best way to get this fungi back is we don't really know how fungi get from one place to another. Like we know very little about dispersal ecology. So it is a huge research gap. So what some other studies have found is that we've planted all this stuff and it's gone pretty well, and then it's got to the later cereal stage, and because the fungal partners haven't been able to get in there, the restoration is kind of stalled and hasn't progressed to the next stage and developed in that way. So there's a great paper that came out just last year called 101 things that we need to learn about dispersal ecology, and it's basically like highlighting that the research gap is 10,000 miles wide. We know very, very little about it. So they'll keep asking me how to put them back, and I'm like, really tricky. We don't know how they move. We don't know how far they get. We've lost a lot of our dispersal vectors through animals as well. And we need to work out how to get them back into our restoration site. So it'd be great to have people who are undertaking active restoration, having a look at things like taking soil cores from adjacent bushland, finding out what's in them through some eDNA sequencing work, putting them in their restoration, and then looking at the survival and proliferation, because we need to be developing some robust methods to get fungi back into these systems. Because a lot of studies, what they're finding is mine site rehabilitation and things is the fungal community is not journeying back to the adjacent reference site. And we need to work out how to ameliorate that. So we need to have a better understanding of what's in soil, what they're functionally doing, and how to put it back. So, okay, so Ros, Sapphire and myself have spent years putting together a great fungi landbook, which Ros is going to have sales. I'm going to plug it today.
And they actually exist now. You can see it.
For $45 each. So if anyone wants to start learning the basics of fungi in a really accessible and friendly way, great book to put on your coffee table. It'll give you the introduction, and then maybe I could get into the deep nitty-gritty of stuff instead of having to go through the basics of stuff. Because there's a lot of nitty-gritty I'd really like to get into with everyone, but if we've only got 25 minutes, we can't quite do that. So what I've done is I've put together a whole bunch of management slides, because obviously it's quite a short talk, that Eve's going to email out that starts to look at the things you can be doing to consider about looking after your fungi. So she'll email those out afterwards. Another thing is I'm going to just bang on about iNaturalists, because I do this at every talk. We don't really have any mycologists in Australia. Mycologists we do have generally do a lot of volunteer work and don't get paid heaps, and we have other jobs. So a lot of our understanding of distribution and dispersal of fungi comes from citizen scientists. So you don't need to know what it is. You just need to be able to take some really good photos of it, call it a fungus, and put it up. And if you can join our Fungi Map Australia group, that's great. If that's too complicated, it doesn't really matter. We'd just like to know what sort of is going in and what substrates. So that will give you those extra fields if you join that project. The big thing though is making sure when you take photos of fungus, I need to see the top of the fungus, I need to see the bottom of the fungus, I need to see the type of the fungus, you know, like the stem of the fungus if it's got one. So you can take more than one photo. You can take 16 photos and put them on iNaturalist. More photos the better. If you can't see underneath, you can get a little hand mirror.
Yes, I've got them.
Ross has hand mirrors for sale today if anyone wants to get photographic fungi. So please, like, it's just most of the significant data papers that are coming out about our fungal ecology is driven by the Citizen Science Project, and everyone can get involved, and it's a great little community to be engaged with online as well. So, cool, yeah.
Thanks, Julie. Right, so that was very, very informative talk. Thanks so much. You guys going to get a lot out of that. So our next speakers is Bruce Ivers, and he is with the Environmental Action Group, and Nikita Freddonet from UWA, and they will be talking about new data on water holding capacity in the soils and Swan Coastal Plain, new ways of looking at seedling survival, but nose. Welcome, Bruce.
Before it starts, could you turn the heat on?
I think it's on 24 too.
Thanks, Vanessa. It's a real pleasure to be here today to be able to talk to you about some of my favourite topics. And are we able to get Carly and Alan off the bottom of the screen?
It's just on the Zoom. I'll just move it around.
Oh, okay, yeah, because I end up with little bits all over the screen.
Yeah, I'll just move it. You did a great job with mine. Thanks.
Okay, thanks, Eva. That's great. So can you take time for the next slide? So I want to be a bit provocative here. Have you run across somebody like this in the Landcare group who's been doing it for 20 years, has the ideas, knows everything, and has probably had 1 year's experience 20 times? Next slide, please. Because what I suggest to you that this is the development of Landcare folklore. And I want to suggest today that we can actually do quite a bit better Landcare folklore. Next one, please. Let's start doing Landcare science so that we're actually looking seriously at variables and trying to tease out what's happening on our site. Next one. So I want to give you an example, and let me just show you where the soil types are, because you know, the land across the top of you. Down the edge here, there's a Quindular series dunes that are yellow colour. If you go inland, it's the two Spearwood series, and then this is Banksia woodland, and that takes up a lot of the Swan Coastal Plain, particularly in the city. So the research work that I've been doing is I've been trying to look at each of those three soil types to see what's going on. So this particular example was to use fertiliser tablets. Do fertiliser tablets make a difference in Quindular dunes? And this was actually done down at Milo. So next one. So that's the trial site last year. And what we have going down the hill, you can see there are lines, bamboo canes, and there's a seedling planted next to each one of those. There are 10 rows, 10 lines, and there's 5 species of plant here. So one row has fertiliser tablets and the other one doesn't. So a group of bushranger cadets from John Tonkin College planted that in the middle of June last year. So next one, and we jump forward a year. Now, just to confuse you, that's taken from looking in the other direction. But sorry about that if it's a bit difficult to figure out, but if you go across to the very left side, you can just see the row of cane going up the hill, the row of canes going up the hill, and that's pigface. And the second row in is pigface with a fertiliser tablet. Wow. Now, the difference is huge. That's a 15-cent fertiliser tablet, made that difference. And some of the others are a bit hard to pick up. This one's quite easy to see. That's Scabola with on the very outside on the right, and then coming back in one is Scabola without. I've done this and reproduced this result 4 years in a row. So I would say to you that on a site like that, and the important thing about like that is that it's bare, because that means it's not water repellent. I've found that these bare sands are quite often they're happy to absorb water, but if you get an area that's already vegetated, it can be quite water repellent. So where it's bare sand, that system works really well. So that's an example of Landcare science, and it's reasonably easy to do. I get school kids to do it, but if you've got a community group, it probably takes a few hours to lay it out and plant it and then watch it over the next year. Go along, do plant counts, see how many survived, how many died, and then you can measure the plants to get an idea of what volume there is. So next one, please, Eve. Seedling survival rates in the wheat belt always tend to be much greater here on the Swan Coastal Plain. And I've been wondering what it is that weed well does that we don't. And sort of emblematic of that is the trowel, because that's something that's very common for revegetation work here on the Swan Coastal Plain. Next one, please. This is a farmer's trowel. So he doesn't have to have a 525 horsepower four-wheel drive pulling that. It just so happens that this one is. And the yellow vehicle is a Chatfield tree planter. It's an easy planter, that particular one. So to get farmers to be interested in doing land care, it had to be done mechanically and it had to be able to be done at scale. So there's a lot of equipment being developed to do that. Next one. Now, the equipment, there you can see the Chatfield tree planter is up in the air. Underneath the drawbar near the front, there's some shiny steel. That's a grader blade that's V-shaped, which takes the topsoil off and puts all the weeds over to one side. So you're not actually getting weeds when it's around the seedling when it's planted. Immediately behind that is a big ripper that goes down into the soil, rips down 30 to 40 centimetres so the roots can get down easily. Behind that is a couple of discs to make a mound so that you've got a ripped soil and a big mound of earth on top of it. And finally, right at the back, there's a couple of press wheels that push down the middle of the mound so that the soil's firmed up again. Let's go to the next one. That's the ripper. You can see the round bit over on the far side of the photo is a disc, but that's a fairly substantial piece of steel. So it's going to go through and rip the soil up. It's got a couple of wings on the back of it, which lift the soil as it goes through the soil, and it just cracks the soil open to allow the roots to go down. Next one. Now, this is off a different planting site, and I actually meant to take it because this is at Wandering, and you can see the frost on the soil at 8 o'clock in the morning. But it also illustrates quite well what the Chatfield rip mark looks like when it's finished. You can see, looking up that first row, there is a seedling there, and he's sitting on top of where the ripper has been. The soil is absolutely ideal for him to grow quickly, to become a big seedling. So next one. Okay, so I thought, how can I replicate that in Kenhurst Park or Blackwell Reach Reserve? They're not going to let me in there with a big tractor like that to drive around. So I have a standard practice, which is a drill with a little 10 centimetre diameter auger on it, and I'll drill a hole about 30 centimetres deep to plant seedlings into. And I thought, well, the other end of the scale from that is to do a huge amount of loosening of the soil. So I used a little ryobi one there. And by the way, these are outside on the table so that you can have a look at them. And the ryobi one is a 15 centimetre diameter auger, and I can push it into the ground 60 centimetres. That's 10 litres of loose soil. I thought, let's go to the extreme. I'll loosen 50 centimetres versus the 2.5 for my standard practice. And let's see if we get any difference. The other thing I found by doing this exercise in that Kenhurst Park site, there is actually a hard pan. There's a hard layer of soil about 40 centimetres down the top. And by using the big auger, I could feel it as I was going in. So we're actually punching a hole through that. And if the plant gets bigger, it's got the opportunity to get its roots down into the subsoil. The other thing I thought was, I've got this really nice little trial, 40 seedlings in each treatment, and that's enough to be statistically significant. Oh, but I really want to find out what terracotta does. I guess everybody's familiar with terracotta. It's a product that has water crystals and fertiliser in it, in essence. So it's fairly expensive. It's probably about 40 cents a seedling to use. But I thought, well, let's just do it. So I did half of the standard practice with terracotta and half of the modified practice just to see what would happen. Next one. And that's a little seedling growing. All the seedlings that went into that trial are Acacia pulchella, so that there is no difference because of different plant species. And next one, please, Eve. And that's the same plant just a few days ago. It's just finished flowering. Actually, can you go back to the previous one for a second? The other thing I wanted to show you was the stake. M on the stake means modified practice, so I know what the plant is. 16 means it's plant 16 out of 20, and a T means it's got terracotta, so that I can go back and easily identify what's happened to each seedling on that side. So plant counts. Forward 2, please, E. Okay, so as a result of doing that, we did some plant counts 3 times. One on the 21st of October, and you can see mid-March and early July. And in essence, the standard practice site had the lowest seedling survival all the way through. On the first time we did the count, the standard practice plus pterococcum and the modified practice had 80% survival rate. I mean, I don't know why, but they were both quite a lot higher than the standard practice. And then if we put the modified practice and the pterococcum together, we've got a higher survival rate again. So it seems like there's some interaction going on there. But look, I have to stress the sample size is too small to draw any conclusions from this. Now, push the button again. Okay, the really interesting thing that came to me from this was the seedling deaths. Look at when they occurred. Between the 11th of July and the 21st of October, we lost 24% of the plants. Now that's during the wet season, and there should be no moisture stress there at all. We go to 21st of October to the 15th of March, we lost another 16%. Well, perhaps. Now I went back through my diary of doing the watering, and I noticed that all of those deaths occurred by about mid-January. There were no deaths after that, even though it was 15th of March when we took the count. Go on to the 8th of July. Look, we didn't lose anything. Now, what's going on there? Look, the best suggestion I can come up with for that is seedlings were probably a bit small when we planted them, and we lost them because they really shouldn't have been planted. But I think I urge you to do counts like this so that you know what's going on your sites when the deaths are occurring. Next one, please, Eve. Okay, this is the big auger that we've used this year. So this is a 20 centimetre diameter auger, and we take that down 60 centimetres, and that disturbs 20 litres of soil. I wanted to have an auger that I could do once for one seedling. We're not disturbing 50 litres of soil like we did last year, but if you're ever going to make this work, you've got to be able to practically, you've got to be able to do one hole for one seedling. So next one. And in conjunction with Mike Ewing, who is a retired agricultural scientist, we devised a trial for next year. And I'm not going to go through all of that, but just to give you an idea, it got a bit complex. There's 10 different treatments. What we were doing was looking at an individual item, like water crystals. What difference does that make to a nil plot? Because both of these, there is a nil plot where I just dug the hole. And then if I just put a fertiliser tablet in, what happens? But what happens if I put a fertiliser tablet and the water crystals together? Does that give me more survival rate? So that's the essence of what's behind this, but it does get a bit complex. Okay, next one, please. But that's Landcare science, and that's what's important. So why bother to do that research? Push the button. Yes. Well, what I'm looking for is disruptive changes. What haven't we thought of that suddenly jumps out? It was a little bit like the fertiliser tablets down at Milo. I never thought I'd get that kind of response. So next one, please. So we look back at Blackwall Reach results from the 24-25 summer. We planted 640 seedlings back at the beginning of June in that year. We only got a 36% survival rate, so that was 230 survived to year one. But here's the interesting bit. I put that together with the volunteer hours, and most of this is watering. So we had 600 volunteer hours. Volunteering WA tells me that a volunteer hour is worth $48.01. And so if you multiply that out, we've put in $28,800 worth of value into that site. Next one, please, Eve. Now that means it's $125 a seedling. That seems pretty expensive, but also I want to bring to your attention that it's important to understand the value that you volunteers, all of you people too, put into what you're doing. Now, let's see if we can change this. Next one, please, Eve. So we'll stick with the same survival rate, but I'll see if I can cut the volunteer hours half. So how could I possibly do that? Well, it might be adding clay into the soil at the time of planting. And the way I would do that is bentonite clay, which you can buy commercially, dissolves fairly easily in water. We have a watering system where we can and pump the water across the site. And we don't use a wand, we actually use a watering spear so that we can inject the water into the ground. We could inject clay in the same way. That's an example of one thing that I could do at the beginning of the year, and it would save a whole lot of time later in the year. That's what I mean by a disruptive activity. So next one, please, Eve. So if we could halve the volunteer hours, we get it down to $73 a seedling. Next one, please, Eve. So if we, on this one, manage to increase the seedling survival rate to the same as that trial that we had at Ivers Park, 61%. And so we're getting it down to $43 a seedling. And final one, if we were to go to wheat belt kind of seedling survival rates around 95%, and again, we've got it down to $27 a seedling, but it still sounds like a lot. But we've saved $100 a seedling by doing some fairly basic things, which also save all of our volunteers a whole lot of time. And so having a group of volunteers who've helped me for a number of years with watering, they're now all getting a bit sick of it, I have to tell you.
How did you?
It's a hard job. So next one, please, Eve. So that lovely person bending over there is Nikita, who was doing some research work for us down at Ivers Park. What we're looking at there was to try and understand more about soil moisture in the Swan Coastal Plain. Next one. And why? Well, we get too many dead seedlings. If we don't do anything at Ivers Park and Blackpool Reach Reserve, seedling survival rates are somewhere between 0 and 5%. So it's just not a proposition to leave them. Next one. So we watered them in. So this was how watering used to be done. It was the fire hose of the Melville tanker and spray it around everywhere. But it made me wonder whether we were actually doing it very effectively by doing that. Next one. So we put black poly pipe across the site and we got garden hoses clipped onto the pipe. And that way you're watering each individual plant. So that makes it a little more effective, but we're still putting it on the surface. Next one. Then we got excited. We got a grant through the Commonwealth Government for planting trees for the Queen's Jubilee, applied for by the Rotary Club of Melville, who've been a great big help to me for years, and I really thank them for their assistance. Next one. And we used the money to put more poly pipe across this pipe. Members unrolling it. Next one. And we got set up with a watering setup like this. There's a pump in the middle that we can pump out of either of those tanks. There's a bore that the City of Melville have given us access to that we can go and fill up so that we can do 3 or 4 tanks at a time if we need to. And we pump it across the site using the black poly pipe. Next one. We got the girls from Santa Maria College to come down. They have a club there called Eco Sisters, and they love coming and doing this. We've just done another planting with them, and 2 of them told me they'd been to all 4 plantings that I run for the school down at Blackpool Reach Reserve. So it's fabulous to see young people that enthusiastic. Next one. And this is a site in the first year that we planted there. We had water and no water sites. So this is a no water site where is it, 4th of September. It was about 10 days before we had the last decent rain in spring of 2023. In fact, the site didn't get any decent rain probably until May of the following year. So that's just before it had some rain. Everything is looking pretty good. Next one. And this is 70 days after the last decent rain. And a significant number of the seedlings were already dead. So that 70 days is kind of giving us an idea of what ability Banksias have got to cope with drought. And also a note up there that 100 days since the last rain, everything was dead. So the question I've got, and I know Nikita's going to talk a bit about this at the moment, is how long do we have to leave between waterings? How far out can we push it? Next one. So this is a site that we watered and the seedlings are looking great. Next one. And that's the same site in November last year. So all of those seedlings have grown up and they look terrific. Next one. Let's have a look at what some of the results we got. In that first year, we got a 10% survival rate, and I wasn't happy with that at all. We planted 1,400 seedlings and we've got about 140 of them through that first summer. And I reckon that an issue is the quality of watering. It's not just a case of going, I mean, he's doing a good job there. He's getting with the plant, but I saw a volunteer who watered one here and didn't bother to water the one over there, to walk over there. He did it from here. And of course, it ended up like this on the ground and it didn't get through to the roots. We were concerned about if we had to improve the quality of the watering. Next one. So what we did was develop some watering spears. Peter White, who's going to talk to you in a minute, came up with the idea from DBCA and Narrogin, and we borrowed Peter's idea, and we're now poking the spear into the ground, delivering the water down to the root system. Next one. That's what a spear looks like close up. There's one outside that you can look at. It's got a flow meter on it, so we know exactly how much water we're putting on. We've put little graduations up the shaft so that you can tell how far you've gone into the ground. And that helped us get from that 10% survival rate up to around about 33 for that site. Not fabulous, but it's a step along the journey of how to make this more effective. Okay, next one. Mike Ewing from Cottesloe Coast Care looked at it, thought that's a good idea, and he developed his own little spear. They don't have to always look like that one for watering along the coast, and he says it's improved his seedling survival a lot. Next one. The other thing we did was we wanted to find out what was happening to the moisture that we actually had in the soil. So this is installing soil moisture sensors at 20, 40 and 60 centimetres below the surface. As Access Tech do this, and the controller up there is buried in the sand, and it communicates with the Access Tech computers via the mobile phone network. So it does about 4 readings a day, I think. And when everything is working well, you can dial in and you can actually just track the water content in the soil, more or less as it's happening. So see that again in a minute. So next one. These are where we installed the soil moisture sensors in the 4 different soil types we have there. Next one, Ed. And this is the sort of graph. I added the green on it, but the blue sections that you can see along the line, the right-hand side of the graph as you look at it is winter time, and the left-hand side going back that way, each one of those spikes is a watering. Now, one thing I discovered is that if you want the sensor to tell you how much watering you're putting on, it's always a good idea to water the sensor. And that's why there's a couple of missing there, because I obviously didn't water the sensor. I watered the plant, but the sensor's a little bit too far from the plant. But what I want you really to get out of that is see in that soil type, which is at Blackwall Reach, how little time the water actually stays in the soil. There's a fortnight between each of those waterings. Some of them was really only there for a day. So the plant, in order to survive, and I didn't think plants could get thirsty, but apparently it's able to take in enough water. If there's anybody here who's a plant physiologist, I'd love to hear the story about how this works. So that's the sort of data we got from looking at the soil moisture sensors. And next one. Now it's time to hand over to Nikita. There she is with Mike, about to take soil samples down at Cottesloe.
My name's Nikita, and I got to work with both Bruce and also Mike over the last year at 4 critical locations across the Swan Coastal Plain. So I was looking at increasing our seedling survival rate, as Bruce has been discussing. The main problems that we hypothesised with both Bruce and Mike was that the Swan Coastal Plain soils had very low water holding capacity. So this is shown in this image of Blackwall Reach, where this is after a lot of rain during winter last year, and we dug up all the soil and it was all completely dry, which meant that all the water had just moved through the soil profile extremely quick. And the other issue that we hypothesised was that there was very high water repellence. So this is an image of Penhurst Park after rain, and you can clearly see that only the surface of the soil is wet and everything underneath has remained completely dry, which means that our rain has not been able to penetrate our soil column. So Bruce touched a bit on this, but I think there's 3 main reasons as to why this research project and even today matters. The first is the ecological impact. So the Swan Coastal Plain is home to threatened ecological communities, the Banksia woodlands, the Chewett woodlands, and also the coastal dune systems. These are all globally recognised as biodiversity hotspots. So it's super important that we look after them, revegetate them, as they are becoming increasingly fragmented due to both urban expansion within the Perth metropolitan area, but also climate change. The second is the financial impact, which Bruce touched on. $125 per surviving seedling is a very high number, and we do need to lower this in order to keep revegetating. And the third one is the social impact. So revegetation relies heavily on community and also volunteer participation, and low survival rates just simply erode volunteer morale and community confidence in restoration. So in order to keep doing this, we do need to boost those numbers up so people feel like they're really making an impact. So I've got to work on the 4 sites that had sensors. The first is Cottesloe on the left, the blue little tag here, which sits on Quindalup Sands of the Swan Coastal Plain. Moving on to Blackpool Reach, which sits on Spearwood Sands with Cottesloe subtype. Just down the road, we have Point Walter, which also sits on Spearwood Sands, but a different subtype. This one's Karrakatta. And the fourth one is Kenthurst Park, locating close to Jandakot Airport. This sits on Bassendean Sands of the Swan Coastal Plain. So all these sites are on very different sands, which means we cannot apply the same management practices across all of them, which is super important and also extremely difficult. So the main objectives of my research that I did over the last year was to quantify the soil hydraulic properties across each of these sites, so different Swan Coastal Plain soils, which I will touch on, to analyse the sensor data that we do have at these sites and develop some site-specific irrigation strategies on how we can increase that seedling survival. I also looked into different soil amendments, such as soil loosening and adding different things to the soil in order to also help boost that seedling survival rate up. So as a quick explanation of what I mean by soil hydraulic properties, or also important plant stress thresholds, here we have a soil column. When it rains or when you apply irrigation, the soil becomes wet and it reaches a saturation point, which is when essentially all of the soil is completely wet. 1 to 3 days after this, once all the excess water has drained, we reach field capacity. So that's how much the soil particles can actually hold on to the water that is in the soil using like suction and their pore spaces. Then our plants use up the water that is within the soil column and we reach the permanent wilting point, which is when essentially there is not enough water for the plants to use and plants will die. This is the point of no return. So plants actually can't recover from this permanent wilting point, and then we just go to when the soil is completely dry, so 0 soil moisture. It's important to note that what we're dealing with here is sandy soils. So if we look at clay, loam, and the blue one is sand, we can see that different soils are able to hold varying amounts of water. Clay and loam manage to hold way more than sand here, which only has a minimal amount. This is because water just drains straight through the soil extremely quickly. So my methods on how I did this was I first went out with Bruce and also Mike. That's a picture of Bruce and I out on the field with an auger collecting some soil samples. I then took some soil samples to the laboratory myself and analysed for water retention, particle size, and also water repellents. And Bruce also brought some soil samples to the CSB laboratories, and we were able to get some chemical properties of the soils we were dealing with as well. I then moved on to analysing the sensor data, which was pretty tricky, and also looked into things like biochar, clay, and also soil loosening to see whether there was scientific evidence of these helping soils in a restoration context. So my first result is here. So these are the water retention curves for the different sands of the Swan Coastal Plain. As you can see, they have a very steep decline. So this is our saturation point. So the point that we reach after it rains. Then it moves straight down to this is our field capacity, which it drops significantly low. And then here we have our wilting point. So as you can see, there is a minimal amount of water that our soils can hold. this is essentially a blown up image of these. So here we have our field capacity, and here we have our permanent wilting point. And if we're to look at the difference between the two numbers, that gives us our plant available water. So we can see that Blackwall Reach can hold significantly more water than Kenthurst Park, for example. So in order to explain this, I tried looking into different things. So plant available water has a very strong relationship with particle size. So as you can see, as the particle size increases, we have the plant available water decreases. So this is because coarser, larger particles, like the ones we see at Kenthurst Park, cause the water to freely drain through it due to smaller, larger pore spaces with less suction. And then we had, so essentially Kenthurst Park had approximately 65% lower plant available water than Blackwall Reach due to its larger particle size, which is why it's important that you also look at your sand grains. The next result was to do with soil water repellency. So I tested all our sites, and Point Walter was mostly the main one that showed soil water repellency, which was pretty surprising because we thought it was actually across all sites. However, to look into this, I read about organic matter content and its impacts on soil water repellency. And a larger amount of organic matter content, which is seen here, we have about 3% or 2% for Point Walter, resulted in a moderate to severe water repellency. However, Blackwall Reach, Cott and Kenthurst Park had an organic matter content less than 1%, which is described in literature as the functional amount a soil needs in order to be functional. So this highlights that Blackwall Reach, Cott and Kenthurst Park could do with the addition of organic matter content in order to make their soils more functional. However, too much organic matter content could also result in high water repellency. So it's super tricky. There's not a set answer, but we've just got to keep looking for the little things that can explain why we are seeing what we're seeing. The final thing that I did was analyse the soil sensor data. So similar to what Bruce had on the screen, this shows the soil moisture content over time. I analysed it and was able to identify the wilting point, which is that dashed line at the bottom here, and also the field capacity point, which is the dotted line at the top. As we can see, when irrigation is applied during the summer months, so that's these little peaks here, and also when winter rainfall comes about, we have about 2 weeks between the water being at a level within the plant available water window and it just dropping under that field capacity point. This means that we need to water about every 2 weeks. The other key thing to note is that the irrigation applied over the summer causes peaks that are a lot smaller than the winter rains. So when winter comes about and you have winter rains, those peaks are closer to that field capacity point, whilst our irrigation is barely getting over that wilting point line, which means we should also probably start applying more water when we do. So this just shows the field capacity and also wilting point for each of the sites. They can't be compared across because unfortunately the soils weren't, the sensors weren't calibrated to the soils. However, if you were to deploy some sensors at your location, you'd be able to identify these points. So you would know that as soon as it drops close to 6, you need a water to get it up to that 28 moisture content again. So in conclusion, I guess what we should do is we should start shifting to sensor-informed irrigation. So if you do have the budget to install sensors at your location, I think it would be extremely beneficial. You'd be able to identify the minimum and maximum amount of water in your soil, and this would allow you to know when you do need to water. Sometimes it could be every 2 weeks, other times it could be 3 or 4, depending on rainfall amounts. Also look into the issues that your site is seeing. So Kenthurst Park and Point Walter could prioritise adding clay, which I think they'll look into doing in order to boost the plant available water and allow the soil to retain more water when it is irrigated. But meanwhile, biochar could be added to Cottesloe and Blackwall Reach due to the low amount of organic matter content we see at that site. In terms of volume frequency, I'd say that the timing that we are currently doing, so every 2 to 4 weeks, is close to correct. However, the amount of irrigation being applied is insufficient in order to get that water content close to the field capacity point across all sites. Yeah, that's
Good afternoon, and thanks for everybody turning up. Peter White, I'm worked for DBCA for 50 years, been in the wheat belt for 35 years, but been working on restoration projects for most of that time. I've also worked in a lot of urban areas, done quite a lot of restoration work with Bruce and Eddie in different places. So this is, I'm here as a more of a volunteer, as well as as well as a practitioner and a community member. So my slightly so to do something better than my riveting title, which was improving seedling, I thought, you know, still bullet a guide to it. There's if we move on to fancy. Here we go. Right, if we so the questions are, do your revegetation look like this, or whether it's an inciting collection of tree guards or stuff that's 5 years old and it's starting to die, or a great collection of weeds, or great collection of nothingness, or the nearly dead, almost dead, possibly should be dead. That's what your re-veg is looking like. The next one, or would you prefer something that looks slightly more lush, got good texture, got wildlife that's moved in? And if you just pause on that one in particular is exciting because it's that's a 1-year-old chewet at Clontarf Hill. So there's this is a very brief overview of things. We haven't got much time, so I'll be rushing through a few things. But I found that I think that the present survival and growth rates that we have in a lot of our urban work just aren't really acceptable. But also, when I've spoken to people, I've found that a lot of the observations that people make of the reasons for failure aren't correct. My assertion is that we actually don't need silver bullets. We just need a consistent, regular attention to all of the detail of a project. So if we move on, these are just some points, keep going, that I'll just be touching on these types of points as we go through, and then you'll be glad it's all over. I work on maybe 95% survival for my restoration work in the wheat belt. Easy for me. I've got a paddock. I've got control over lots of things. But yes, when people cite 10 or 15% survival, that does worry me greatly. Yeah, people have become, I think people have started to accept that that's normal. Whereas I think we really do need to challenge it. And by the way, what I'm talking about will be a fairly basic prescription. What I'm putting across is what I think is Re-Veg 101. This is not Rolls-Royce in any shape or form. This is just attention to the details as we go along. So again, planning for success. I mean, who plans for failure? But if you're working on 15% survival, that means I think you're working on 85% failure. So we've already started to plan for infill. So we haven't started to plan for success. We've started, we know we're going to fail. So we're ordering our seedlings already. So That's a somewhat jaundiced way of looking at things, but and I saw an example recently where at a nursery, somebody came in, grabbed a heap of seedlings. They grabbed the leftover seedlings at the nursery. They were going to bung them in because they got some money. So you think, where's the planning? Where's the preparation? Where's the thought? It's just a quick response. We need to plant seedlings. Here they are, in they go. So we focus a lot on the doing. We don't focus a lot on the legacy and the outcome and being able to go back. By the joy of working in the week for 35 years, I can go back to trees I've planted a long time ago. And most of these things that I'm telling you of what not to do, I can probably say that at some stage along the line, I've done them. So I'm speaking from some experience. So I'm planning for, I'm planning for legacy, which is, just imagine the site in 30 years' time. That's really what you've got to try and focus on most. I'm planning for the project, which is the big picture stuff, the species, the quantities, the layout, the weeds, the logistics, all of that type of stuff. Do you have the time and do you have the resources? Do you have the people? Do you have the intellectual clout to plan a project? You know all of the stuff that you need. And then you're planning for the day. Is the soil moisture adequate? Have the weeds been controlled? Do you have enough supervisors to make sure that things go in green side up? It's all of that sort of stuff. There's a great emphasis on the community planting day, but that's not the end of it. That's only a very small portion of our revegetation work. And if you click on the last slightly facetious one, this is what I think we apply to our work. And I try and tell people, seedlings do not live on hope. We thrive on it, but our plants apparently don't. So when we looked at the cost of failure, just looking at this, if we looked at 2,000 seed planted, 80% failure, $2.50. We've spent $5,000 in the first year, 80% survival. We'll come back and we'll put another $4,000. And just if that process continues at 80% failure, by the time we were at year 5, we've outlaid $16,808. $8.40 a seedling planted, 6,000. You look at the time and the effort and the volunteer exercise or costs, and that's why I think we need to recast these things. We don't talk about 15 or 20% survival. We actually say it as it is. It is 80% failure, and I mean that's a bit harsh maybe, but it's, I think, the way we have to look hard at things. If we go to the next two, that's my first rendogram of the presentation. But we couldn't fit it anywhere on the slide any other way. But if you look at the top left-hand corner, the site inspection, the getting out, not a desktop, getting out and having a look. And then you have a look at where below it is the seed collection, the organising of the nurseries. Because if you go in January with a bag of seed, the nursery is not going to be very happy with you at all. If you go in June and say, oh, have you got some seedlings I can have? Well, it's going to be slim pickings. So you've got to have a timeline like this where you work out when you need to start, because some species you might have to need to collect the seed the year before. And also when you have a look, once you've finished your planting, you have to avoid the temptation to nick off on holidays for a month or so, because your surveillance of your planting site should start the week after you've planted. You need to go, because once the weeds start, if you come back in a month's time, it's like, geez, I've got weeds. Well, by that time, it's usually too late. So next, if you look at the design, and we'll talk a little about species choice, layout, structure, biodiversity considerations and amenity considerations, because this is the legacy. This is what's left. What you've got to look forward to is watching how this develops, and so improving survival will the species choice will help improve survival, because people often look and say, well, I planted things that were from the reserve, but your planting site might only be a very small part of that reserve. So you need to have the species that are appropriate to where you're going to put them in. So that can have effect on seedling survival. Your layout, and I'll show you a few pictures of this, the layout of things, I see far too many plants planted underneath the crowns of existing trees. And so your seedling survival is affected by your layout. Your structure, rather than just fling everything in and hope that whatever comes up works, I tend to use sometimes single species to link, single species to provide particular habitats. So I try and create a whole lot of diverse habitats across the site, and I think I get better survival rates, because if you put everything in and you've got a whole heap of Acacia saligna, then that's going to grow really well, and you're not going to have much left underneath. So you need some sort of segregation of your seedlings and placement of seedlings across the site. And there are some amenity considerations. If we go to the next, this is a little bit of Clontarf Hill. You'll see that there's some there that is unplantable because that was standing bush. You'll see where we put mixed species. The dots there are of where I put the chewet in, so that there's enough room between the standing trees and they're not underneath. They're not underneath canopy. I've buffered areas. I've put in much more distinct habitat plantings, and the amenity strip around the edge is so that we've got some nice taller ones, because we've got patches of shade that go up along our pathways. So you can get better survival out of your plantings just with the right placement of seedlings. And if you go to the next one, design does matter, because that's a poor species choice. The site wasn't evaluated correctly. After 5 years, all of the Melaleuca hamulosa decided it wasn't salt tolerant. And that's a significant portion of it. So working out your species and your sites are important. The 2 clicks, there's a seedling being planted right up under the canopy of another. 2 clicks more, and there you have another example of it. Put a lot of effort into planting things in places where they're not going to live even if we water them. So yeah, just so the simple layout of plants, which is really can be difficult at a community planting day. You have to be the ones that go and put the plants out where you want them to be. Seed, this is where it all starts, and the legacy is your seedling survival. You're talking about longer term things here rather than just survive to the next year. if you're using seed that's been collected from a planted tree down the end of your driveway, you know, collecting from one plant that's inbred, that's genetically compromised, your seed source is really important, because it's the long-term survival, getting good and good genetic mix, and just also thinking that seed is so incredibly scarce that we want to make every seed work, and the 15% survival or the failure rate means that we are churning through huge amounts of seed for very little return. Seedling quality, we'll just click through why it's important, why we would care, how we can achieve it, some key issues. We'll talk a little bit about trays, standards, and the implications of poor quality nursery stock. Good quality seedlings, so much easier to handle, whether it's from transporting, whether it's actually the physical planting of them. The survival maximises the use of volunteer time, and it's, yeah, look, one of the things with this is I've had the value of going back to many sites, and some of the things you're about to see, when I saw problems with seedlings, when you try and do diagnostic work and people say, oh, drought, something like that, something meaningful. When you actually go out and dig up seedlings, that's when you start to find what the real causes of plant death. So we'll have a look at some of these. Now, some of these, next one. Yeah, look, if you're lucky to find some of the seedlings in there, and there's a few, that's probably the poor quality seed that you've given to the nursery quite late. Next one was a few collections of moss and some waterlogged things. However, if you keep on, whether you would prefer this, there's some examples of well-grown, topped, robust seedlings, good density, good vigor, no weeds, very regular, very uniform, good colour, and so on, at the right height as well, both minimum and maximum, and so on, and just say the uniformity of it. So they're the sorts of things that I like to plant. If we go to the next one, if we look at seedling standards, you can make a request for particular container types, things like the position that the seed is in, whether it's sown or if it's transplanted, how it's transplanted, whether it needs to be thinned, it needs to be graded, whether it needs to be topped to be the right height. So all of these are specifications where you can go to a nursery, you can talk to nurseries and say, this is how I would like my seedlings to look, because we know historically that these sorts of things, when you have a height range, when you have one season old seedlings, not stuff that's been in the back of the nursery for a couple of years, properly developed root systems, 64 per tray or whatever it is, and that they're healthy, disease-free. You are the customer, so don't accept substitutes of things. And I do a nursery inspection 3 times a year, so I know what I'm going to get, so that you don't have, the nursery turns up with a wheelbarrow full of seedlings on planting day and said, sorry, mate, you have to be the ones that go in there and check and inspect so that you know how things are progressing, so that you maintain a control over your project. One of the things, if you have a look, the effects of seedling containers on plant root systems is a really big issue. You can see on the top left there, the coiling and the pots underneath, and the shape of the pots and the way that it induces the root curl. And the ones on the right there are open weave trays, and there's some examples of those out the back. Next. And so the ones that don't have any treatment, you get this mass of uncontrolled roots. On the root trainer pots, they're much more contained, and the ones with the air pruning, you can see that the roots are much better shaped, much better developed. Also, with all of these, particularly the last one, they actually fill the entire soil bolus there. There's quite a bit of argument on whether you should plant forestry tubes or cell trays. The point's not necessarily the type, it's whether or not the roots are adequate, where the top is adequate. It's actually the quality of the individual plant rather than the container. But I've seen people with sort of slavish dependencies on forestry tubes, but I haven't seen any evidence to say that they're any better than anything else. The seedling root development, here's a couple. You can see how you've got off-centre seedling roots, and sometimes unthin. And people say things like, you would think that once it's in the ground, those roots will take off. Well, if you preface anything you say with you would think that, you've identified a gulf between that and the reality. The reality is when I go and dig those up, or you've got really bad, you've got really bad J-rooted seedlings, or you've got really bad coiled seedlings, they're the ones that I find. And if we keep going, because we'll have to flick through this pretty quickly, lots of little coiled seedlings, that's a badly transplanted seedlings. You can see next one, all of the mini plugs, and you can see just horrendous problems. And this is so the implications, you get sudden death in the first year, you can get reduced growth rates, compromised growth rates, and plants more susceptible to wind throw. What's wind throw is when your trees blow over. And the next, that's a seed production area. That's root binding that's caused that whole seed production area to fail. That is a 2008 seedling, which I put out of its misery. And the next one, that lived for about 18 months. Yeah, it was planted as an advanced seedling, but that's what you get with poor quality stock. Yep, that's poor twisted thing. The survival count, briefly, I worked out that I think the trees started to decline even when they were watered. They started to decline because the ones with the poorer root system just could not adapt. So it was really important. More problems. Okay, so we've got no roots. We've got no roots. We've got too many. We've got things that are too tall. We've got wispy things. We've got unthin things. So, look, seedling establishment, weed control, feral animal control, site prep, mounding if necessary, planting time, planting quality, post-planting, protection. Right, I'm going to go through these pretty quickly. Keep going. There's no such thing as a few rabbits. Weed control. Yeah, there are plants that are just rabbits with those. This is a bit tongue-in-cheek, but I don't care what equipment that you have, just as long as you use it And even if you've only got a hoe, it's making sure that all those weeds are depriving your seedling of moisture. So hang on, Tico. Zoomed out. So the consequences, you know, where are they? That line on the left got mist spray. So here's your post-planting. Now there are seedlings in there. And so what's been killing your trees? Your site preparation techniques do a good job of that. We've planted in salt. We've not attended to weeds. The hoons have got in. They didn't get planted properly. It fell out of the planting bucket. Or it's a very poor fertilizing technique. What's been killing our trees is actually the usual suspects. Now, we'll just go through quickly, plant the shortcut to successful tree planting. The planning, the seed and seedling quality, site prep, vermin, weeds, fencing, basic stuff. Start planning for the new site, because there's a couple of things to leave you with. One is when you stop and evaluate what the single biggest cause of seedling failure is, us, because we didn't control the weeds, we didn't pick the site correctly, we didn't do all of these things, we didn't attend to the detail. And that's something that we really need to have a look at. My suggestion is, as far as silk bullets go, if you've got a werewolf problem, that's fine. If not, give them back to the Lone Ranger. And just thinking about these next ones, where you have a look at how that site's developed, how the structure of this, the survival rates, and the growth rates that we've got, and we're back at this point again. And the next one is my favourite little cartoon, because the single biggest cause for success has got to be us, because it's our project. This is what we know how to do. This is what we should be doing. So with that, I hope the gods of revegetation smile upon your project.
It's been good. I've followed on from the presentations of the other speakers to reinforce what we really need to look at in terms of seedling quality. I get the pleasure of working with those nurseries that you sometimes love and sometimes criticise, and that's the role that I have in Green Life Industry Australia. So we look at how we can support the nursery industry to produce the quality seedlings that you require. Yeah, thanks. So the Australian plant production nursery industry, there's about 1,600 nurseries across Australia that are called production nurseries. So they're not retailing, they're not selling direct to the public like a garden centre. They're the nurseries that grow the bulk of the plants for Australia. I produce seedlings of all species across all climate zones, of course. Similar problems are experienced by all growers at different times, and that's a fact of life, that we all have exactly the same issues. Having been a nurseryman for 40 odd years, I've done everything wrong that you could possibly think of, and have tried to come through it. But my role now as an extension officer is funded by the Nursery Industry Container Levy, so it's industry funded. It's not government supported at all. It's the industry that pays for the extension service that we're looking to produce high quality plants. We promote best practice management through continuous improvement to achieve the high health production of nursery plants, and the research funding is provided by Hort Innovation, who administer the fund, and by the committees that are part of Hort Innovation. The whole Australian wholesale nursery industry turns over about 2.8 billion, so it's only a small part of the agricultural type production. but it's, we think it's pretty special, and that was from February 2026. So the Australian Plant Production Standard, it's been developed by the growers. It's not a research-based standard at all. It's backed by research. The website's available for all the growers. You can go and have a look at this yourself. You won't be able to get access to all of the sites unless you're a paid-up member. It's Nursery Production Farm Management System, fms.com.au. It's been around for about 35 to 40 years, and it's a massive repository of information. So the app examines the inputs to growing plants, so potting media, water and irrigation system, propagation equipment, plant health protection, and the management systems by growers. And the aim is to produce high health plants for your produce. Thanks. The limitations, and Peter's covered a lot of these as well, and we can see them from the other presenters as well. Seasonality, of course, it has definitely affected us. I mean, this year we're looking at a low winter rainfall year. It happens because where we live, rainfall patterns, drought, wind and storms, etc. The sites, the variation of the soil type, etc. Seedling availability, nursery capacity this year has been really strained. There just aren't enough producers around. And there's been some issues, again, looking at the variability of seedlings. Seedling quality and survivability, they're directly related. And how do we go about assisting the purchasers? And the funding cycles for community programs, I'm sure I was speaking to the converted about that you are constantly fighting against availability of funding types. One of the main parts about that, of course, is that nurseries are in business. They're there to make money. So they need that supply to come through earlier rather than later. If you have trouble ordering your seedlings on time, then that's going to be reinforced with the availability of seedlings when you need them, of course. Lack of time for planning and communications and understandings. Site preparation, of course, we know about pot to container size. So we deal with the seedlings through from the cells right through to the landscape tree type product, the 35 litre through to the 500, 700 litre containers. And so depth, volume, root trainers, etc., and that root shoot ratio. Thanks. So setting the expectations. As I said, the nurseries are in business. They need to know what you want out of the end product, and you need to know what you want to set, what you want to ask the supplier to produce for you. Do they line up? Sometimes not. The expectations can vary quite a lot. So that's a skill that we all have to sharpen ourselves up, and that's the negotiation skills, where you need to direct the business that's running or growing your seedlings, what you want the plant to look like when it comes out the other end. Set up communications channels with the grower. As Peter was saying before, he visits the nurseries, roughly 3 times a season. Do that, please. Go and see them early. They need to be talked to so that you get those expectations balanced. Spread the order over at least 2 growers. That's called risk management. Everybody know about risk management? It's a pretty important tool. And set the milestones for inspection and review related to payments. Again, those nurseries are in business. If you go and have a look at the seedlings and you've already formed a contract with them, with the grower, that they will receive a payment for a certain stage, I can guarantee it'll be done. That's seasonal conditions included, of course. Agreement on substitution. Don't take anything. Make sure that you've got a plan for list A and list B, and look for the nursery accredited businesses so that they've been through the standards and the sub-programs. We have a range of about 18 different sub-programs under the accreditation side of things. The landscape tree stock specification. There's another one called the ethical nursery stock specification as well. Accredited nurseries do work within them, and that's part of my job as well, to make sure that they stay within them, and they're assessed every year for that. Ensure plants are grown in root trainer pots. We really need to work hard on that. Even for the bigger trees, they need to be root trainers. Root architecture is absolutely critical to survival. Peter's shown you some great examples of what not to look for. So that goes right back to the seed side of things and the actual training or the root preparation for transplanting. Direct seeding is definitely best, straight into the pots. So I've lost my place. No weeds. Yep, weeds compete directly with the seedlings, so make sure there's no weeds in them. Check on them early. The nurseries that are growing under the apps, they are required to actually set up a monitoring program through the growing season. And that's another aspect that we look at. Set the plant seedling specifications with the grower early. What do you want the plant to look like? You know? Any suggestions? One stem, straight up, 5 sets of leaves, you know, a certain diameter, hard stem, maximum minimum height take for average height. We've seen every trick-in under the sun plate on average height. Set the minimum height and the maximum height, and set the delivery and receival timeframes when you're expecting the seeds. Again, seasonal conditions change every year. And document and record the agreements. Really important. So you've got some comeback. Again, you're in a contract. You're in a purchasing contract with the business. There's only a few community nurseries that are so-called, not for profit, but it's quite a different setting. So they're for-profit businesses. Thanks. Timing the process. Timing is everything. Site preparation essential. Understand the limitations of the sites. Order the seedlings and set the specifications. You need to do it early. Check on seed availability. Seed shortages, we're only just beginning to see that. It's going to get considerably worse as we go along because areas to collect seed is just diminishing. Availability or access to those areas, unless it's done illegally, is a big problem. So just make sure that you set the species and seed availability. We needed to be putting in seed production areas 25, 30 years ago. Did we? Not very well, but we need to be looking at that. There's still time. We've still got another 25 to 30 years ahead of us to do that, which will be really good. Review the seedling germination with the nursery. Bad seed batches or poor germination numbers are pretty common, depending on how it's collected, etc., and handled. Modify the order early if required. And don't forget to prepare the planting site. Pretty important to get that done early. Know the limitations. Thanks. Site preparation, weed and pest control. Know, again, document what's on site. Hardened or acclimatized seedlings prior to delivery are really important. That process needs to start roughly April, thereabouts. I'm just picking a date out, but roughly April. Pre-drill the holes. We've seen the value of disturbance. We know that disturbed sites actually get a much higher survival rate. In the plantation industry, we look at a minimum of 95% survival. That changes considerably with community planting, so it's to do with disturbance and preparation. Pre-drill the holes or pre-rip the holes or whatever. The disturbance of non-wetting soils is absolutely critical. It makes a huge difference. Ameliorants or adjuvants such as clay. We've revolutionised agriculture in the south coast of Western Australia by adding clay. We've doubled the yields on thousands of hectares of farmland simply by adding clay. Because of mining machinery now, you're shifting thousands of tonnes of clay. It's quite a simple process now. It costs, but to double your yield on agriculture is quite revolutionary. What's happened in the last 15 to 20 years. Planting, ensure the root seedling cell is fully wet at planting. That means more than field capacity, actually make them completely soaked. And make sure that we keep the water up to them on the planting day. Don't let them dry out at all. Root compaction at planting. full contact, no air pockets, tamp them down hard. There's a planting machine that's working in the southern part of, well, over most of the southwest land division now. It actually has 2 pistons behind the planting tube, and it jams the soil in. You can't actually pull the seedling out. The percentage of survival has rocketed up from sort of 80% through to 98, 99% because of that tamping. It's a simple mechanical process. You can't do the same thing with your heel, because you don't have that hydraulic pressure, but it just goes to show that a few changes have made a big difference. Protection from predators, that's 2 legs and 4 legs. We see theft of plants in particularly urban plantings now is a very real problem. And maybe those silver bullets might need to be. I couldn't go down that road. Irrigation at or after planting, really essential. Our planting window has collapsed completely, particularly in the Perth metro area, with the inability of the soils to hold moisture. So we're coming down in planting window. Again, with machines that do that tamping, the window's actually opened out and they can start planting on sort of 25 mil of rain in whenever that is, in March or April, if that's the case. It can go through until you actually can't move on the site. Yeah, thanks. Yeah, some questions a little bit later on.
Look, thank you so much, everyone. And thanks to our speakers. We've had a fantastic afternoon.